Alright, so in this lecture, we are going to take a look at protein synthesis. So we know that protein synthesis, or at least this will become familiar to you soon, that we synthesize proteins from a strand of mRNA, and we get that strand of mRNA from DNA. So we'll undergo this process of DNA to RNA to protein, and we'll term this process the central dogma. This is sometimes called the central dogma of molecular biology, and it's an extremely important topic in molecular biology because proteins direct so many bodily functions as enzymes, as catalysts, as receptors, as well as many supporting structures. So a lot of our muscle is composed of protein, and it's very ubiquitous and very important for our bodies. So this central dogma of what I'm talking about, we go from DNA to RNA to protein. You could really come in here and draw an arrow of DNA going back to itself because we know that DNA will replicate through DNA replication, DNA synthesis. DNA will make a copy of itself using a template, and then using one strand of DNA as a template, we'll then make an RNA strand, and this ribonucleic acid will then be used and transcribed into a protein. So this process of DNA to RNA is called translation. I'm sorry, this is called transcription, and then RNA to protein, this is going to be called translation. So you're transcribing something first, and then you're going to translate RNA into a new functional protein. Okay, so the way we do this is we take a section of DNA um, that is termed a gene, and this gene is a segment of DNA that is going to be used specifically to make a very particular protein. Okay, so there are sections of DNA all throughout our genome, all of our entire base pairs in our DNA. There are certain segments that will be used to make a particular strand of RNA, and then that will be used further to make a specific protein. So this is important for us. This is important for other animals. Genes regulate um, bodily processes. It directs development as we are developing as a embryo and into a fetus during our earliest stage of development. So let's take a look at this process a little bit further. Like I was saying, transcription and translation. These are the two parts that we'll look at. We'll look at what happens here, and we'll look at where they happen as well, because these two processes occur in different parts of our cell. So to understand this process of protein synthesis, we have to review the structure of DNA. So we remember that DNA is a double helix. It contains two strands that are twisted around each other. We know that it contains a phosphate group, it contains a monosaccharide that is a deoxyribose, and it contains four different nitrogenous bases. It contains adenine, guanine, cytosine, and thymine. Okay, A and T always pair together. C and G always pair together. Okay. We take this DNA and we will transcribe it into a strand of mRNA. So as we look at this, we'll remember and we'll go back to this 5' end and this 3' end because there are very specific enzymes that will function in only a certain direction. Okay, when we make our new RNA strand, we'll substitute in a ribose monosaccharide for a deoxyribose, and we'll swap in a uracil for our thymine. Okay, and generally our mRNA and our RNA is going to be a single strand as opposed to our DNA, which is a double helix. So these are the two differences, at least two of the differences between DNA and RNA, the sugar as well as the nitrogenous base, deoxyribose versus ribose, and thymine versus uracil. So the first stage of protein synthesis is going to be called transcription. Transcription, if you think of something being transcribed, you're just writing down what's being said, or you're writing down something that you're seeing. And so that's exactly what we're doing. We're taking DNA and all we're doing is we're just writing it down in the form of RNA. Okay, so everything will be the same except we'll swap in uracil for thymine, and we will use a ribose sugar instead of a deoxyribose sugar. Okay, what we use is we use a strand of DNA. So we untwist, we undo the helix of DNA, just a segment of it. We use one strand as a template, and we can use that one template strand to then make our new um, RNA strand. So let me go back. Whenever we are making our RNA, what type of RNA are we making? Well, we know that we have mRNA, rRNA, and tRNA. The type of RNA that we're making at this stage, transcription, is the mRNA, so messenger RNA. Now, after we make this messenger RNA strand, what's the difference between this DNA template and the RNA strand? Well, that's what we just said. We differed in the sugar, and we differed in the nitrogenous base. Ribose sugar versus deoxyribose, uracil versus thymine. Okay. Now, all of this is occurring within our nucleus, and this should make sense because where is our DNA? Where does it reside within our cell? Well, it resides within the nucleus. So in order for transcription to occur, we have to do that in the nucleus, and then we know that we will leave the nucleus through nuclear pores to enter into the cytosol, where we can undergo the next stage. An important note moving forward. We said that a gene was a segment of DNA that's used to code for a specific protein, but to do that you have to get to an mRNA, and then that mRNA strand is going to be read in our next step, translation at the ribosome. It's going to be read in series of 3 bases. So we know we have adenine, uracil, cytosine, and guanine. We're going to read it in 3 base pair segments, and one segment that's 3 base pairs long is going to be termed a codon. Okay, so a codon is on the RNA strand. We'll also see a term called an anticodon. That anticodon is going to be on our tRNA, or our transfer RNA molecules that are also attached to an amino acid. So this process of transcription, what we're going to do is we're going to take an enzyme. You can break it down into 3 main parts: initiation, elongation, and termination. Our first stage, we're going to take an enzyme known as Rna polymerase. Okay, so RNA, we're dealing with an RNA molecule, and we're polymerizing. So if you go back to some terminology, we know that a monomer is something that is just a single unit. A polymer is made of many single units, so more than one. So a RNA polymerase is going to polymerize DNA. It's going to, polymerize RNA, and it's going to make this long and lay down all these strands to make this long RNA strand. Okay, what it does is it binds to a promoter sequence. It needs a place to start, essentially, that is found on our DNA, and from there it can then begin this process of inserting appropriate bases and inserting appropriate nucleotides to our growing mRNA strand, and that process is termed elongation. So we started it with initiation. We're now beginning to elongate this mRNA strand as RNA polymerase is laying down lots and lots of nucleotides with their nitrogenous bases, so with adenine, uracil, guanine, and cytosine. After it transcribes that entire gene, that section of DNA, it will encounter a stop codon, and that will tell transcription to stop. Okay, so transcription stops, RNA polymerase unbinds. We now have our finished mRNA product, and we can move on to translation. There's actually a small step that can occur, however, between transcription and translation, and we'll take a look at that in just a second. So here's a more visual representation of what's happening during protein synthesis. We have a RNA polymerase, and we have our DNA here in red, and we have our growing mRNA strand here in green. Now, I referenced the directionality of DNA and the directionality of RNA at the beginning of this lecture. So if we look at this strand, that this red strand right here where my mouse is, this is the DNA template strand. If you follow this and you follow it up and you follow it down here, this is the 3' end, and likewise if you follow it this way, this is the 5' end, and you know the direction that RNA polymerase is moving. It's moving from the 3' end to the 5' end, okay? So RNA polymerase has a 3' to 5' direction to it. It moves from the 3' end to the 5' end, and it lays new bases down, new nucleotides in the opposite direction. So instead of it laying it in the 3 to 5, it lays it from the 5 end to the 3 end, okay? So a kind of phrase that you can use to help you remember this is read up, going up 3 to 5, you're going up a number. Read up, write down. So we're writing down the numbers. So we're writing from the 5 to 3 direction, okay? You're reading up 3 to 5, you're writing down 5 to 3. That's the case for RNA polymerase. It's also the case for DNA polymerase whenever you get to DNA synthesis. So RNA polymerase moves from 3 to 5, writes from 5 to 3, and we're using one DNA template strand to make one mRNA strand. Note the bases. You have C and G's pairing, and you have A's and U's pairing. Note that there are no T's in the growing mRNA strand. You have T's in the DNA because that's fine. You can have T's in the DNA, but there are no thymines in our growing mRNA strand, only uracils. Okay, so here's another picture showing the same thing. So what you can see here, if we're using this bottom blue template, this bottom blue DNA strand as our template, and this is the 3' end and this is the 5' end, you can begin to understand which end the RNA polymerase is moving. So if we read up and write down, we're reading up from this way, so that means we'd be going from left to right here, and we're writing down, starting at the 5' end, and now we're laying new bases down at this 3' end here. So we'd be moving from left to right here. So this is what I briefly alluded to a second ago. There can be a small stage in between transcription and translation that you can call post-transcriptional modifications or some type of splicing that goes on. And so what you have is your finished mRNA strand. There are some sequences of bases in there that are non-functional and not necessary for the functioning of that mRNA sequence to be able to be translated into a protein. So what you have is you have splicing. You have introns and you have exons. Introns are those that are not required, they're not needed, they will be spliced out and removed, whereas the exons are important and remain after the splicing occurs. So what you have is introns taken out, all the exons will then join together to make your finished final copy of your mRNA. So here you can see everything in blue is an intron. It is non-useful, so those will be spliced out by enzymes called spliceosomes. They'll come in and splice those out. And then you have all of your exons left. They will join together to make your final mRNA strand that will then be used for translation. So where are we going now? We're going to go to the, if you think about where we started within the nucleus. We have to go through the nuclear pore to get out to our cytoplasm. And it's in the cytoplasm where we'll undergo our next step of translation. So we'll stop here and we'll pick up with the next part in the next